Welcome to the third week of the course. In this week, you will learn what is the biological profile of an individual and see the methods to estimate the biological profile of non-adults. First of all, an important thing is that in human osteology, we will always talk about estimation and not determination of the biological profile. This is because we can't be certain about these characteristics of the individuals unless the ones that belong to identified collections and have that information. The biological profile of an individual is a fundamental tool to understand a population. The biological profile includes the estimation of age at death, the sex, stature, ancestry, and morphology. We'll just delve into the methods about age at death, sex, and stature, because these are the ones that are almost always done when studying a sample. But I will talk a little about the other characteristics. To estimate each of these informations of the biological profile, there are scientific methods, and the identified collections are very important to develop these methods. Before we start to talk about the methods to estimate the biological profile of the individuals, the choice of them is important. These methods should always be adequate to the population studied, meaning that, when possible, the methods chosen should have been developed with a similar population. This is because there are biological differences between different populations. For example, some populations have a medium stature higher than others. There are several methods to estimate the ancestry that are based on the characteristics of the skull, especially in the face, given that it is the place where the differences are most seen. And this is because we have adaptations to the climates that we live in. This image is part of the method of Effner, and it analyzes various characteristics of the face. In this case, you can see the differences in the nasal aperture. You can see here that this one is narrow, this one is medium, and this here is broad. Together with other characteristics analyzed, the method gives us a probable ancestry. The methods to estimate the ancestry are usually more used in cases of forensic anthropology. In archaeological contexts, these methods are usually applied in cases where there are suspicions that those individuals are from another ancestry than the one from that country. I left you this method and another one in the supplementary materials in case you are interested in this topic. The morphology consists in the physical characterization of a population or an individual. We can do this with metric and non-metric traits. The metric traits consist in measures in specific bones and places of the bones that gives us information, for example, about the biomechanical activity, and consequently can give us information about occupational activities. The stature is included in the metric traits, but we will talk more about stature in the next week. The discrete characters are small variations in the skeleton of an individual. When analyzing these traits in a population, sometimes it is possible to associate with certain places. For example, the Inca bone that we taught last week. The individuals that have this bone can be from the Inca population. I left you a book that is a photographic atlas of these non-metric traits if you're interested in seeing some of them. Moving on to the biological profile of the non-adults. In the non-adults, the biological profile of an individual consists only in the age of death estimation. The sexual estimation is more precise only when the skeleton hits maturity, because it is during adolescence that the differences between females and males are developed. There are some methods that have attempted to estimate the sex in non-adults, but they aren't regularly used. It is, however, possible to estimate the sex in non-adults using the DNA. The stature estimation can't also be done because the non-adults are still growing. As it is implied, the estimation of the age at death in skeletons is relative to the age that the individual had when he died. There are two kinds of age, the biological and the chronological age. The chronological age corresponds to the number of years lived since birth, and the biological age is characterized by the conditions of the individual. It is dependent of the aging of the different parts of the body, varies between individuals, and the factors that influence them aren't constant during life, and it is influenced by the environment. When we are studying a human skeleton, we are estimating the biological age. When we estimate the age at death, we don't get an exact number, but yes, an age range, for example, 12 to 18 years old, or an age class, for example, young adult. 7 age classes and ranges commonly used to segregate human skeletons are from Buikstra and Ubulacker from 1994, and these are fetus, before birth, infant, 0 to 3 years, child, 3 to 12 years, adolescent, 12 to 20 years, young adult, 20 to 35 years, middle adult, 35 to 50 years, and old adult, more than 50 years. Grouping the individuals in these age classes is very important to have a global view of the population. This way, it's possible to see which age class is most represented and least represented. These age classes are also important because they allow comparisons between them. For example, we know that the diseases have a higher impact in children and older individuals. In times where there were no medicine, these were the ones that died the most. In the case of a non-symmetry from a deadly disease, using the different age classes, it is possible to compare the number of individuals of each class and see the ones that were the most affected. To estimate the age at death, it is important to understand the biological and physiological processes, both development and degenerative, that affect the human skeleton during life. The processes of development are used to estimate the age at death in adults and the degenerative changes in adults. The development of the skeleton consists in the growth of an individual. As I said last week, for example, the fusion of the epiphysis to the diaphysis. The development of the human skeleton has a big genetic control, so is less susceptible to environmental factors. Because of this, the estimation of the age at death in an adult is more precise. The degenerative processes consists in the changes that occur to our skeleton after we get to the adult age and start to get older. They are highly variable due to environmental factors such as nutrition, occupation, and activity, and this makes the estimation of age at death in adults less precise. The age at death estimation in an adult is based on the changes that occur due to development and growth of the skeleton. I'm going to present you methods based on the dental eruption and calcification, the epiphyseal union, and the length of the diaphysis of the long bones. There are several methods for each of these processes of development, but I'm only going to show you one of each, the ones that I think are the easiest to understand and more used. First, eruption and calcification of the teeth. This method is a more precise one, given the eruption and calcification of the teeth is under tight genetic control. This way, it is the method that gives us an age more closely to the chronological age. When using a method to estimate the age at death by the teeth, it is important to note all the aspects of development, including the completeness of all crowns and roots, and the place of the tooth relative to the alveolar margin. The alveolar margin is this zone here that is basically the place where the teeth fully erupt. It's the limit between the crown and the root of a fully erupted tooth. I'm going to present you one method commonly used with the teeth from Ubulacker from 1999. This method provides this image here that represents the dental development in gray, this teeth in gray, are the deciduous teeth, and in white, these ones are the permanent teeth. On the left side is the age corresponding to each stage, so this age corresponds to this stage. And it is only represented half of the inferior and superior dentition. You can see that in each stage the teeth have different sizes, sometimes only the crown or the crown and the part of the root. You can see here this teeth is complete and here this one is not. And as you can see here, this line is the alveolar margin, which means that when the teeth are superior to this line, they are erupted, and when they are inferior to this line, they haven't erupted yet. When applying this method to different teeth, it can reveal different stages of development. In these cases, it is possible to make a narrow interval, but I'm going to show an example. In this image, you can see a mandible. It is the same mandible, but in two different views. And this place is here without teeth. It's because they were lost post-mortem, so they were lost after the death. Now we are going to see the teeth. So first we have the permanent canines, and they are permanent because they haven't erupted yet, but we can see the size of the roots. So according to the method of a Blacker and using the canines, this individual have an age range of 4 to 9 years. You can see in the image that from 3 to 9 years the crown of the canines is already formed. But we are starting on the 4 and not on the 3 because the crown of the tooth is already visible. And on the ages before 4 years, it would be impossible to see so much of the crown of the tooth. And if you look at 10 years, the inferior canines are already erupting, so this can belong to the age range. And then we are going to see the first premolar. The first premolar is completely erupted, and it is a deciduous tooth. According to the method, from 3 to 9 years is when the first deciduous premolar is fully erupted. At 2 years, this tooth isn't fully erupted, and at 10 years, the first deciduous premolar no longer exists. And then the second premolar. The second premolar is a permanent, and you can see that it isn't erupted, and we can also see the size of the root. So according to this tooth, the individual has between 4 to 10 years. At 3 years, the crown of this tooth is very small, and at 11, there are deciduous teeth. And lastly, we can also see the first premolar that is permanent, that is also not erupted. Again, we can see the size of the root. And with this tooth, this individual has between 1 to 5 years. At 9 months, the crown is too small, and at 6 years, this tooth is already erupting. So, now that we have information from every tooth in the mandible, we are going to combine this information. We can do a simple graph like this, where we put every age range. Then we see the smallest age range that results from all the others. And as you can see here, the smallest age range is from 4 to 5 years. So this individual had an age from 4 to 5 years. The fusion of the postcranial epiphysis is orderly, and the epiphysis fuses at a known age. To this method, I am going to refer to a book that is called Development Juvenile Osteology from Schuer and Black from 2000. And this book has all the ages that every epiphysis fuse. This scheme here represented is only a summary. It should be noted that the fusion begins earlier in females than in males, so usually each epiphysis has 2 age ranges. To use this method, it is important to know when an epiphysis is fusing. In this image, you can see 3 femurs, and you see the proximal epiphysis. In femur number 1, the head of the femur is infused. In this second image, you can see this line here that is called the fusion line, which means that the head of the femur was fusing when this individual died. And then, in the femur number 3, you can see here that the head is completely fused. So for this method, we only use the bones that have the fusion line. So, using the second femur as an example, we consult this image and see that the age range that corresponds to this epiphysis. In this case, the age range for the females is 13 to 18 years and for the males 14 to 20 years. Given that in the majority of times we don't know the sex of the non-adults, as in this case, the age range goes from the earliest age to the older, including both the ages of the females and males, giving a final age range of 13 to 20 years. As well as in the teeth, the same individual may have different age ranges from different epiphyses. In these cases, we make a graph like the one from the teeth to find the smallest age range. Before we see the last way of estimating the age at death of non-adults using the length of the diaphysis, it is important to know how and with what the bones are measured. And when we are referring to measures in human osteology, we talk about osteometrics. That is the study of measures of the skeleton. There are several tools used in this discipline, but I'm only going to show you the ones that are used to the methods that I'm presenting. The first tool is the sliding calipers, this tool here. It has a pair of jaws whose gape is measured via a scale on the caliper shaft. This tool is usually used to small bones and small measurements. And then we have the osteometric board, which is this instrument in here. As you can see in this image, the board is composed of a long horizontal plate with an attached perpendicular end plate, here and here. And then a graduated skull extends from the fixed end plate, and there is a second movable end plate whose position can be read from the graduated skull. And this tool is used to measure big bones. Finally, this method is based in the length of the diaphysis of the long bones. That is, it can only be used when the epiphyses aren't fused. The method has this table that it is where we take our conclusions. You can see that it can be applied to the humerus, radius, femur, and tibia. To each bone exists a medium value of length and a range of values of length. After the measure is taken, the value obtained is compared to the table of the method, and we see in which age range this measure fits. Then, we only have to see the corresponding age to the age range. I will give you an example in the next slide. Depending on the size of the bones, the measures are done with the sliding calipers or the osteometric board. To give you an example, we have this femur. And as you can see, none of the epiphyses is fused, so we can use this method. Let's say that this femur was measured and the length of the diaphysis is 307.5 centimeters. With this, we go to the table in the column of the femur, in the mean value, and we are going to see where this value fits. And as you can see, this measure is between the ages of 12 and 13 years old. So the age of this individual, based on the length of the diaphysis of the femur, is 12 to 13 years old. As well as with the last two methods, different bones can reveal different ages. The multiple method approach consists in using different methods to estimate the age at death. In the case of the non-adults, the methods commonly used are the dental eruption and calcification and the fusion of the epiphyses. In this way, there is more information and the age range is more precise. In the next week, we are going to talk about the biological profile of adult individuals. If you have any questions, please let me know, and I will see you next week.